So what you can see here, this is a spinal cord. So everything outside of it, this is going to be the meninges, right? And this is the spinal cord itself. So how do we call this center right here?
Central canal.
Very good. So this is a central canal. Which kind of cells are going to be lining it?
Ependymal cell.
Very good. Ependymal cells are lining it, and this is where the CSF is going to be located. What is this?
Dorsal horn.
Very good. This is the posterior or the dorsal horn, and this one right here, this is the anterior or the ventral horn. What can you find in the dorsal horn?
Soma of interneuron.
Very good. So majority of the interneurons are going to be here. And in the anterior horn, these are the cell bodies of who?
The alpha motor neurons.
Alpha motor neurons. Yes, guys, you're right. Okay, so these little dots that you can see here, they are very big, right? They're even visible with this magnification. So these are going to be the cell bodies of the alpha motor neurons, ventral motor neurons, right? Or lower motor neurons. They're all the same. Everything else here, this is the white matter, right? So how do we call this area?
Dorsal
Funiculus.
This is going to be the posterior, what?
Fasciculus.
Not fasciculus, but
Funiculus.
funiculus. Fasciculus is individual tract, right? So fasciculus is one tract that could be here, but the entire area, this is known as the funiculus, yes. What about this area?
Lateral funiculus.
So this is going to be lateral funiculus, and the one which you're going to see here, this is going to be the anterior funiculus. Yes, that's true. Okay, so we have mentioned majority of the things. How do you know that this is the anterior side, though?
fissure.
Artery.
So first fissure or the sulcus, right? So here, what do we have?
Anterior fissure.
So anterior median fissure, and here this little tiny invagination, that's the posterior median sulcus, right? And somebody said vessels. So this structure right here, this is the anterior spinal artery. This one right here is the posterior spinal artery. As you can see on our slide, we can only see one of them. There's just one of them that is going to be here. Another one should have been here, but it isn't. Due to the sectioning, we cannot see it. Okay, so we're going for meninges. So let's start. What is this?
Dura mater.
For sure. So this is a dura mater. How do we call this space right here?
Subdural space.
Yeah, so this is the subdural space right here. Epidural space is above it, right? It's going to be right here. Okay, so this is where the epidural, it's above it. So epidural, subdural space. That means that the second thing right here, this is
arachnoid
matter. That means that the space here is
subarachnoid
space. Yes. So subarachnoid space, this is where the CSF will be, right? So this is the location of the cerebrospinal fluid right here. If this is the subarachnoid space, that means that this area right here, this is the pia mater, right? So this is the pia mater right here. What is this? A part of the pia mater.
Denticulate ligament.
Very good. This is a denticulate ligament. It's very thick, right? How many denticulate ligaments were there?
21 on
Very good.
each
21
side.
pairs. And what was the purpose of denticulate ligaments?
Compartmentalization of CSF. And
Perfect. Yes. Okay, so compartmentalization of the CSF and stability of the spinal cord, right? So it keeps it stable right here. Okay, how do you call this? So this is pia mater. What is this?
External glia limitans.
External glia limitans will be in the brain, and it will be right under the pia mater.
Oh,
Okay, so we
okay.
won't be able to see it very nicely here. I'm talking about the space specifically. What is this? It's very easy, guys. It's just an artifact. Yeah, so this is not how it should be. The pia mater should be on the surface of the spinal cord. It should be right there. So this space that you can see here, this is an artifact. This is not a sub-pial space. Okay, so you can see where we are, where the ventral horn is going to be, right? And you can notice that in this ventral horn, we can see a bunch of huge cell bodies. These cell bodies belong to the alpha motor neurons. What is another name for them, for these cell bodies, for these alpha motor neurons? They are lower motor neurons. Lower motor neurons innervate what exactly?
Skeletal muscles.
So they are responsible for innervating the skeletal muscle. Who tells lower motor neurons that you can see here to do so?
Upper motor neurons.
And where are they located?
Brain to spinal cord.
So in the brain, okay? So they're going to be in the brain. So in the cortex of the brain. We'll talk about it next class, but it's going to be in the cortex of the brain. You remember the other name for upper motor neurons? They were also known as the Betz cells, right? So we also call them Betz neurons. They are the upper motor neurons. Okay, you can see that there are big, do I have? No, I don't have. You can see that they're big cell bodies, and then right next to them, there are little ones, like this one, and this one, and this one. Do you see them? There are lots of little neurons right next to them. Which neurons are these?
Renshaw inhibitor.
Very good, yes. They're Renshaw inhibitory interneurons. They were located in which lamina? Which lamina was here?
9.
Very good. So Renshaw inhibitory interneurons are sitting in lamina number 9, and they have one purpose, right? So actually, my bad. Lamina number 9 is all the way here. Okay, so that's lamina number 9, and they have one sole function. The function of Renshaw inhibitory interneurons is to control the upper motor neurons, right? So remember that our movements are very nicely coordinated. So the reason for that is that we don't have jerky movements. It's Renshaw inhibitory interneuron. If alpha motor neuron gets too excited, that will cause too strong of the contraction. So Renshaw will inhibit it, right? Renshaw will inhibit it. Okay, how do we call this triangular area at the back? Do you see? So this is the sulcus right here. It's a very, very little deepening right here. So how do we call this triangular part?
Comma tract of
Schulze, very good. Okay, so this is a comma tract of Schulze. Comma tract of Schulze has another name. It is also known as the interfascicular fasciculus, because you remember that we have lots and lots of tracts here. Another name for tract is fasciculus. And you can see that this comma tract of Schulze is sitting in between everybody. So this is why its other name, it is also known as the interfascicular fasciculus. Why is it so red? Why is it so like that?
It's less myelinated.
Very good. It is less myelinated, right? So the axons that you see here, they do not possess as much of the myelin. Okay, how do you call this area right here?
The funiculus gracilis.
Very good. Fasciculus gracilis, and the one right next to it?
Cuneatus.
Cuneatus. Together they are known as?
The dorsal column.
Posterior column or dorsal column tract. You remember that this tract is the major sensory tract there is, right? So this is the one which carries the most of the sensory information all the way to the brain. Gracilis carries from where?
Lower limb.
Very good, right? So because it is located medially, so it has to be carrying it from the lower limb. The cuneatus will be from the upper limb. You remember the border?
T6.
Very good. It's going to be T6, yes. Okay, so everything below T6, this is the gracilis. Everything above T6, this is going to be V cuneatus. Okay, so this is your central canal. You can see that these cells right here are ependymal cells. They are more or less simple columnar epithelial cells. And you can see that they possess cilia and microvilli. Like it's very random, do you see? Some cells do have cilia, some do not right next to it, right? Some do, some do not. So these cells that do not possess the cilia, they possess the microvilli. So it's correct to choose in the exam that ependymal cells of the central canal of the spinal cord possess both cilia and microvilli. So some cells have cilia, some have microvilli. Okay, are you looking at white or gray matter?
White,
Very good, it's a white matter. How do you know this is a white matter?
because of the myelin axons.
Yes, okay, so you can see that this is the axon, right? So this is the axon itself, the white thing, this is the myelin. I agree with you that we can see lots of axons, but don't forget that in the spinal cord, the gray matter also possesses axons and dendrites. Okay, so it also could hypothetically have axons and dendrites. So how do you know that this is exactly white and not gray?
No cell body.
Perfect. No cell bodies. You do not see any cell bodies. Whose nuclei are these?
Schwann cell.
Schwann cells are in the peripheral nervous system. This is a spinal cord, so
Oligodendrocytes.
very good. These nuclei are nuclei of oligodendrocytes. So every single, well, not every single one of them, but majority of the big ones, they are nuclei of the oligodendrocyte, because don't forget, right? So spinal cord, CSF, central nervous system. Okay, so here you cannot have the Schwann cell. It has to be the oligodendrocyte. Yeah. Okay, we have spinal, never mind, I forgot. I go back here. Or here, okay, let's stay here. Okay, tract, I'll just quickly point and you'll tell me which tracts are going to be located there. Which tract is here?
The dorsal corticospinal tract.
Very good. So just not dorsal, lateral, because dorsal is for the cerebellar ones. Okay, so for the spinocerebellar one, there is dorsal and there is a ventral spinocerebellar. There is a lateral and anterior corticospinal. Okay, so this one right here, closer to the gray matter, this is the lateral corticospinal tract. Anterior, as you remember, is going to be somewhere here. Okay, so this is where anterior corticospinal tract will be. What was the job of the lateral corticospinal tract?
For the limb.
Yes, okay, so it was innervating the limbs, right? So anterior corticospinal was innervating the trunk, right? Skeletal muscle of the trunk. Right next to the lateral corticospinal tract, there is something else here.
Look the spine.
Amazing, Ryota, amazing. Okay, free class today, very good. Okay, so this is a rubrospinal tract. Okay, so right here is going to be rubrospinal tract. And as you remember, it's responsible for what?
For crawling.
Okay, so definitely free class today. Definitely. Okay, so you proved it. I swear you proved it. Okay, so yes, it is definitely for crawling. So you remember we have discussed this, that until you're 1 year old, you're using approximately all of your limbs, right? So this is why it's rubrospinal tract that innervates that. But lateral corticospinal tract is just for the limbs after you're 1 year old. Okay, speaking of spinocerebellar, right? So you remember the dorsal column tract, right? So it's right here, the sensory, right? So it's right here, the gracilis, cuneatus, sure. Spinocerebellar. So there's going to be dorsal one right here, and there's going to be ventral one right here, right? So that's majority of the, like that's the major ones that you need to know for the exam, right? So dorsal column, lateral, anterior corticospinal, rubrospinal, and spinocerebellar. Yeah, okay, so spinocerebellar. Okay, then let's go for this one. So we have Bielszowski impregnation. Bielszowski impregnation is just silver, as you remember, right? So it's nothing fancy. It's the same as silver. We just call it with a fancy name. We call it Bielszowski. So the same idea, right? So central canal. How do we call this area?
Lamina 10.
That's where lamina 10 is located. I agree with you, but this area has a name.
Gray commissure.
Very good. Yes, so it's called gray commissure. Okay, so this part right here, this is a gray commissure. That's the area that is joining the two parts, right? So the dorsals are here and the ventrals are going to be here. Okay, so this is the coma tract of Schulze. Sure. What is this? You see, it's very
Lissauers.
yes. Okay, this is the Lissauers tract. Yeah, this is the Lissauers tract, is the one which is kind of giving all of the sensory input to these laminas right here. So you can clearly see that the purpose of this slide is to definitely show you these laminas because they stand out. Do you see? They do not impregnate the same as the rest of the tissue. So these laminas are lamina number 1 and 2, right? So you have to remember there. Okay, so here, if we talk about meninges, there is no dura mater. Okay, so dura mater was lost during preparation. So you can actually see the arachnoid and the pia mater. Okay, so subarachnoid space and subdural space is above it. So dura mater was lost. This area right here is the Lissauers tract, right? So you can see how the axons are coming in this way. And this is the area of the dorsal horn where you can see the two laminas, lamina number 1 and lamina number 2. If you compare the two laminas, you can see that lamina 2 like does not impregnate whatsoever, right? So it's almost nothing there. Why is that? Because here we have almost no myelin. Okay, so lamina 2 is this jelly substance. You remember we call it substantia gelatinosa, right? It's basically like a jelly. And this jelly is a little bit free of myelin. So as a result, you know, it does not impregnate very well. Okay, so it doesn't impregnate very well. What was the job of substantia gelatinosa? What was it doing?
Pain and temperature perception.
Very good, pain and temperature. In lamina number 1, we had a nucleus. It was called?
Posteromarginal nucleus.
Very good. And its job was?
Also pain and temperature?
Just pain. Okay, so temperature is only in laminar 2. Okay, so only substantia gelatinosa is capable of actually sensing or understanding the temperature, right? Or better said, conveying the temperature all the way to the brain. You remember which substance was in the substantia gelatinosa?
Roland.
Rolandos and?
Substance P.
Very good, substance P. Okay, so substance P is pain. Okay, so it's not only going to be in lamina number 2, but also in lamina number 1. This slide is to show you the alpha motor neurons, right? And you can see that there are some kind of cell bodies right next to it. Okay, so these are going to be the Renshaw inhibitory interneurons, and you can clearly see that these Renshaws are really do you see there are lots of like axons going in all the different directions, so there is communication happening. Okay, so you can see that this is like dendrites, right? And there's another Renshaw, another alpha motor neuron, so there's definitely synapse happening. So if you kind of spend a little bit more time here, you will actually see how the axons and dendrites of the two different cells synapse. Okay, so the bigger one are the alpha motor neurons, The smaller ones are the Renshaw inhibitory intraneuron, like this one. Okay, Golgi. Golgi is also silver, but double silver. So Bilushovsky is 1 dose of silver. Golgi is double dose of silver. So the difference between the two stainings is just the amount of silver that you have used. That's it. The purpose of this slide is to differentiate between axons and dendrites. So this is why, if I can advise you, spend some time here, okay? So spend some time, try to really see the difference between axons and dendrites. Because whenever they want you to identify an axon or a dendrite in the exam, they will take you to this slide, okay? So the picture will be taken from here. So let's take a look. You can see that this is a cell body. So what is this?
Axon.
Take a look, it's branching, do you see? Do you see it has a little bit of branches? It starts thick, you follow it, it gets thinner. It can be an axon. So this is actually a dendrite. This is one huge dendrite right here, okay? What about these ones? Also dendrites, you can see that they're branching out, right? These ones, dendrites branching out. Do you see there are lots of dendrites? Lots, because you remember how multipolar neuron looks like, right? So we're in the spinal cord, we're here. We have interneurons and we have motor neurons. Inter and motor neurons are multipolar neurons. So they have a cell body, one axon, and a bunch of dendrites. So if you actually compare, what would you see more, axons or dendrites? For sure, you will see dendrites more because they're just multipolar neurons here everywhere. So it's very difficult to actually find a good axon, okay? So it's very difficult, like you can see dendrites, 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 it's all dendrites. All of them here are dendrites, right? So let me try to find one. Okay, so this could be, so this little tiny thing, this could be an axon. It's very thin, consistently thin, and there is no branching. These things are not coming from it, they're a bit behind. Okay, this could be an axon because it's kind of consistent. There aren't any branches there. Let's take a look here on the next slide. So dendrites, 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 right? So you can see it's almost impossible to find an axon. But of course they will. Guys, I'm sorry, I can't find you an axon. That's dendrites. Okay, maybe, no, that's also dendrites because they're branches. Dendrites, dendrites, dendrites. Okay, for the next class, that will be my homework. Okay, I'll go back to this and I'll just spend some additional time and I'll try to find you a good axon, yeah? So for sure, there will be some axons here. Okay, guys, so this is additional slides that you guys have, but they are not going to ask you on the self-control test, okay? So these slides are just to show you how big the alpha motor neurons are. There are different sections, okay? And you can see that alpha motor neurons are definitely the ones which stand out. So they're the biggest ones in the spinal cord, okay? It won't be in the exam. It's just was there to show you them, but that's it. Okay, spinal cord is finished. So let's move to the next one. And the next one is going to be cerebellum. Okay, so we're going to talk about cerebellum. Share screen, this. Okay. So cerebellum. Okay, so cerebellum is also known as the little brain. Okay, so there is cerebrum and there is cerebellum. So cerebrum is of course the bigger brain. Cerebellum is the little brain. It is located posterior to the pons and medulla, right? So it's kind of behind it. Between them, between the pons and cerebellum, you can see that there is a cavity of some sort here. This cavity, this is the fourth ventricle. Okay, so this is the fourth ventricle right here. That's where the cerebrospinal fluid will be. Cerebellum is located under the occipital and temporal lobes, right? So they're kind of resting on the cerebellum. And cerebellum has a very important job because it controls muscle coordination, posture, and equilibrium. Okay, so this is the job. Now, I want you to understand that it's important for motor control, but it does not initiate the movement. It doesn't start it. Okay, you simply control the movement, but you don't start it. Where does the movement start? It starts in the brain, in the motor gyrus, right? In the motor precentral gyrus. That's where, we'll talk about it next class. That's where the motor thought initiates. That's where we start the idea that we need to move a certain muscle. But these axons of bed cells, they will have something to do with the cerebellum. Cerebellum will be aware of the strength of this thought of yours to move a muscle. So remember that it's more like a motor control. In the self-control test, they use a word that is called fine-tuning. There you go. Okay, so fine-tuning. Do you know what fine-tuning is? Like you fine-tune a musical instrument, yeah? Like you like for piano, you have to press several times to make sure that the string is very much like aligned. So you constantly have to be doing something. So cerebellum is never really silent, right? So it's constantly is fine-tuning all of the action potentials from the brain to the spinal cord. It makes them better, yeah? So that's what it does. So not only motor cerebellum has something to do, but also the cognitive functions such as attention, language, fear, pleasure. Okay, so all of this is also going to be related to cerebellum. You don't have these slides because I recently have added them. Okay, so it's just, it will be in the video. It's just very important for you for the anatomy. That's it. Okay, so it's not for histo, it's for the anatomy. You have to remember that cerebellum consists of 3 lobes. You can see that this is the anterior lobe, posterior lobe, and floccular nodular lobe. For embryo question, you have to remember floccular nodular is the most embryologically untouched. So this lobe right here is the same since you're an embryo. There's no additional structures were added there. Yeah, so you made it during 9th week and you stay with the structure right now. It's the most primitive, it's the most embryologically untouched lobe. Okay, so its job will be very, very primitive. It's not involved in the higher functions because it's so primitive. Yeah, you still have it since you're an embryo. So remember that. Okay, so floccular nodular, as you can see here. So most importantly is that if you slice the cerebellum, it kind of looks like a cauliflower. Okay, so you will see that if I go back, it does have this particular cauliflower appearance. The outer matter is the gray matter and the inner matter is the white matter, right? And you can see that cerebellum also possesses gyra, right? You can see there's some kind of deepenings are also present there. But the gyra of the cerebellum have a different name. They're going to be known as the folia. So this particular protrusions of the cerebellar matter, they are known as folia. So folia are gyri. You remember gyri? We talked about it in the previous lessons. Gyri are in the brain. So this little tiny convolutions of the brain matter, they are called gyra. The same thing in the cerebellum is called folia. Just the same thing, just different name because it is cerebellum. In the anatomy, you will be learning some of the names of this particular folia, but they aren't important in neurohisto. Okay, we have talked about that. Okay, so let's take a look. So this is the histological representation of the cerebellar tissue, right? So we have the outer area is going to be the gray matter, and the inner area is the white matter. The gray matter, as we have said, is the outer part, so it is cortex. So we can say that cerebellar cortex consists of 3 layers. The most outer layer is the granular layer. Then we have the Purkinje layer, and then we have the molecular layer. Okay, I'm so dumb. It's the opposite way. Okay, so it's the opposite. So granular is here, Purkinje, and this is the molecular layer right here. There are total 5 neurons in the outer cortex. Okay, so here we have the molecular, Purkinje, and the granular layer. But in the outer cortex, you can see that there's going to be 5 neurons. These 5 neurons are stellate, basket, Purkinje, granule, and Golgi cell. So for the exam, you have to remember that you will not find these cells in the medulla. Medulla is the white matter. It's the deeper part of the cerebellum. These neurons are present in the gray matter, in the cortex, and they are distributed like that. In the outer area, in the molecular layer, you will find these 2 neurons, stellate and basket cells. In the second layer, in the Purkinje layer, you will find Purkinje cells. In the inner layer, you're going to find the granule and Golgi cells. So out of these 5 neurons, only granule cell is going to be excitatory. The rest of them are inhibitory. Could you remind me, inhibitory neurons, which neurotransmitter do they use?
GABA.
GABA or
Glycine.
glycine. You're going to see that here in cerebellum, these 4 neurons, right? So the stellate, basket, Purkinje, and Golgi, they are GABAergic inhibitory interneurons. All 4 of them are going to be using GABA to inhibit something. The granule cell is going to be excitatory neurotransmitter, but it's not going to be using acetylcholine. Okay, so it's not. We're going to see that it's going to be using glutamate. Okay, so it's glutaminergic excitatory neuron. So does it make sense, right? So 5 neurons, outer cortex, 4 are inhibitory, 1 is excitatory. So could you tell me, in molecular layer, which cells?
Stellate and basket.
Stellate and basket, and they are
Inhibitory.
inhibitory. Purkinje layer possesses Purkinje cells, inhibitory. Granule layer possesses granule and Golgi, right? But Golgi is inhibitory, granule is excitatory. The white matter, the core of the cerebellum, it has a different name. There's a Latin name for it. It is also known as the lamina alba. Okay, so white matter of the cerebellum is also known as the lamina alba. Okay, in the cerebellum, you have 3 very important structures. They are called peduncles. Okay, so you can see it here that this is cerebellum. This is pons, right? The kind of in front of it will be pons, and here we have medulla. So there are 3 peduncles: superior, middle, and inferior peduncles. Peduncle is just a fancy way of saying tract. Okay, it's a bit bigger than a tract because it contains more than 1 tract. Yeah, so peduncle is like combination of tracts. There are lots of axons traveling there. Superior cerebellar peduncle is the main output to the cerebral cortex. Okay, so if we have axons traveling from the cerebellum to the cerebral cortex, these axons have to pass through the superior peduncle. But they also have to stop in the thalamus, because next class, we're going to see that nothing reaches the cortex without thalamus knowing. You remember thalamus in the center of the brain, right? It's a major nucleus there. So if, I repeat, cerebellum needs to send an axon all the way to the cerebrum, this axon will be traveling through the superior cerebellar peduncle to the thalamus, to the cerebrum. Yes? The middle cerebellar peduncles has everything to do with pons. Okay, it's the major tract that simply connects pons to the cerebellum. Okay, so that's what it does. There's so much connections between pons and cerebellum. This is why middle cerebellar peduncle is the biggest of the three. It contains so many different tracts or fascicles. We will talk about them in a bit, right? So middle is the biggest one, and its connection is cerebellum to the pons. Inferior cerebellar peduncle is going to be a bit lower, of course, and it has input from afferent fibers from 4 places. Okay, you're going to see that I wrote here 3, but there are going to be 4 places. It's going to be vestibular nucleus, spinal cord, tegmentum, and medulla oblongata. Okay, so 4 areas are sending their fibers to the cerebellum through the inferior cerebellar peduncle. So if I'm in the spinal cord, right, and I want to send an axon to the cerebellum, right, you're going to see that I'm going to go through the inferior cerebellar peduncle into the cerebellum. So to see that you understand, let me ask you this question. In which peduncle will the axons of the spinocerebellar tract will be?
The
Inferior.
The inferior, right? Because inferior has the connection with the spinal cord. Very easy. In which peduncle pontocerebellar axons will be?
Middle.
Middle. Okay. Thalamocerebellar or cerebellar thalamo tracts are going to be in
Superior.
So do you get what I'm trying to tell you? I'm trying to tell you that this area is like a division. Okay, you have to make more or less classification, right? So you can't just send all of the axons to cerebellum and that's it. So you kind of divide them. It depends on which area of the CNS are you connecting with. Okay, let's talk about superior cerebellar peduncles. So last class, have we learned this word or no? Because I don't remember if I mentioned it. Have we talked about it? No? Okay, you remember I always tell you there's crossing of fibers. Yeah, so in neuro, this crossing has a name. It's called decussation. Remember, for example, lateral corticospinal tract on the left, right? It's responsible for the right arm, right? Or right leg. But the fibers were crossing at a certain region. So this crossing in neuro has a name. It's called decussation. So you're going to see that the tracts which are within the superior cerebellar peduncle, okay, the ones which are within here, they're also crossing. Do you see? They also come from different areas. They also have to cross. So from the right side, it goes to the left side, right? So from the left side, it goes to the right side. They aren't showing it, but that's what's happening. So it's going to be decussating. Where is it decussating? At the level of inferior colliculus. Okay, so inferior colliculi, I'm afraid to ask, you remember last class or no? We have superior and inferior colliculi. They had something to do with the visual and auditory input, right? So inferior colliculi are going to be exactly here, okay? So right here where the decussation happens, that's where the inferior colliculus will be. The tracts that are going through the superior cerebellar peduncle are creating cerebellothalamic tract. Okay, so the axons which are going through this cerebellar peduncle, they're creating cerebellothalamic tract. But there is additional tract that can also be there, and it is called cerebellar rubral tract. Because cerebellar thalamic tract is going to the thalamus, but cerebellar rubral is going to the red nucleus. So there are two tracts that are passing through the cerebellar peduncle. Does it make sense? Cerebellar thalamic and cerebellar rubral tract. What exactly thalamus and red nucleus do, we'll talk about it next class, but for sure you can understand they have something to do with movement, right? So thalamus, red nucleus, rubrospinal, right, corticospinal, for sure it has something to do with nucleus, with movement. Now, there is a particular tracts that are these cerebellar thalamic and cerebellar rubral tracts. They're creating a horseshoe shape commissure, because you're going to see that in addition to these tracts kind of traveling within the superior cerebellar peduncle, the rubrospinal tract is also going to be coming down in this way. Okay, so rubrospinal tract is also going to be coming down in this way. So do you see that these tracts, so in this area, this is the cerebellar thalamic, right, and the rubro-thalamic. They are this area, they're this part. The rubrospinal tract is going to be down, it's going to be this part, it's going down like that. What is the shape? What is this shape? This is the horseshoe shape. Do you see? It's like this. It's the horseshoe. Do you see this? So this horseshoe is called horseshoe of Wernicke. And horseshoe of Wernicke consists of this tract that we just mentioned, right? So on one side is going to be the ascending, the ones which are going to the thalamus and the red nucleus. On another side is going to be the descending. And this horseshoe of Wernicke, it's very important because lesions, right? So problems, lesions, some traumas above it will cause contralateral cerebellar signs, but lesions below it will cause ipsilateral. We shouldn't get too much details in it because that's 5th year neurology, okay? This is by Short-Zolli. It was given by Short-Zolli. So that's why, will it come on the self-control, on the exam? I highly doubt, I highly doubt. This is higher neuro, yeah? This is like 5th year neuro. We're gonna be talking about this particular tract, but he did mention it, so that's why I put it here. But it's Short-Zolli, everybody knows, right? So he talks about some things that are like unnecessary completely. So just for the future, in histo, it won't be asked for sure. This part, it won't be asked. Just in the anatomy, if you get cerebellum, yeah? If you get cerebellum, maybe with Short-Zolli, and he asks you some kind of horseshoe, that's what we're talking about, yeah? So this is the horseshoe right here. If there is a lesion above it, right, there's something problematic above it, it will be contralateral. If the lesion is below it, like in medulla or somewhere, it will be ipsilateral. That's the one thing I want you to understand. Is it clear? Okay, middle cerebellar peduncle, we have said that it has something to do with the pons. We have talked about that this is the biggest peduncle out of the 3. So it possesses 3 tracts. You don't have to know this information right here. you have to know that there are just 3 of them. They're called superior, inferior, and deep tracts. These tracts are just traveling between cerebellum and pons. That's it. Okay, so what is exactly the function of the pons? We'll talk about it next class. But for now, just remember that there are just 3 tracts that are going between pons and cerebellum. These 3 tracts are sitting in the middle cerebellar peduncle. Inferior cerebellar peduncle has a very interesting appearance because it is a rope-like. Okay, if I go to the next picture, here you go. Okay, so do you see it's like a it's like a rope that is crossing on the other side. Would you be able to see this in the anatomy? No. Would you be able to see this in histo? No. This is a theoretical question, yeah, that it is a rope-like structure. The most important thing is that this inferior cerebellar peduncle compromises a structure which is called a restiform body. So this area right here, this inferior cerebellar peduncle, you could say its other name is restiform body. That's its other name. Everything which is right next to it, so like a nuclei of some sort, they are called juxta-restiform body. Okay, so there's some nuclei nearby, you're gonna call them juxta-restiform body. Juxta-restiform is not really important. Okay, so restiform is important. Why? Because the axons which are running here belong to a very particular tract. Which one? The posterior spinocerebellar tract. You remember there's anterior, posterior? So posterior spinocerebellar tract, it will be, the axons will be there. They will be in this restiform body. Oof. Okay. Now I'm gonna talk about how the cerebellum works. So you remember that we have 3 regions in the 3 layers in the cerebellum, right? So we have the molecular layer, we have the Purkinje layer, and we have the granular layer. The most important, all of this is the gray matter, you remember, right? So this is all the gray matter. The most important neuron in the gray matter of cerebellum is a Purkinje cell. You remember we have said that Purkinje cell is sitting in the Purkinje layer, right? But do you see that that's technically not true? What is the one thing which is sitting in the Purkinje layer?
Cell body?
The cell body of the Purkinje cell. Only the cell body of the Purkinje cell is sitting in the Purkinje layer. The dendrites of the Purkinje cell are sitting in the molecular layer, and axon is sitting in the granular layer. Is it clear? Right? So you can see it's a one huge neuron, and this one huge neuron sort of spans through the entire cortex of the cerebellum. Purkinje cell is interneuron. Okay, all of them are interneurons here, by the way. Okay, all of them. So Purkinje cell is interneuron. So what is its shape? It is multipolar. So it has lots of dendrites and one axon. Purkinje cell is excitatory or inhibitory interneuron.
Inhibitory.
Very good. And as you can see, what is it synapsing with? What is the Purkinje cell synapsing with? What is it inhibiting? It is inhibiting deep cerebellar nuclei. There are 4 of them. We'll talk about them right now. But these nuclei relay information to the thalamus from cerebellum. Okay, so these nuclei have something to do with movement. To simplify it, if Purkinje cell does its job, if it inhibits these nuclei, there will be no movement. You won't move. Because thalamus and cerebellum, they actively participate in that, okay? So they have some kind of function in that. So again, Purkinje cell is interneuron, it is inhibitory interneuron. What does it do? It inhibits deep cerebellar nuclei. These nuclei participate with, they participate by interacting with thalamus. Thalamus interacts with cortex. So if cerebellum does not interact with thalamus, thalamus will not interact with cortex, there will be no movement, okay? So there will not be any movement. So to simplify it, if Purkinje cell does its job, if it inhibits deep cerebellar nuclei, there will be no movement. Remind me, what was the function of cerebellum?
Muscular control.
It was motor control. It was fine-tuning the movement. So keep that in mind. You can see that in addition to the deep cerebellar nuclei, we have here additional nucleus, which is called inferior olivary nucleus. Inferior olivary nucleus has also something to do with movement, but it doesn't want to inhibit the movement. It's the opposite, okay? It wants to actually stimulate the movement. It wants the movement to occur. So it sends axons to stimulate the deep cerebellar nuclei. The axons are called climbing fibers. Okay, climbing fibers, you can see they're going from the inferior olivary nucleus all the way to the deep cerebellar nuclei as well. But climbing fibers are called climbing not because of that, because you can see that some climbing fibers from the inferior olivary nucleus climb the dendrites of the Purkinje cell, right? You can see that they're interacting with the cell. Climbing fibers are excitatory, okay? They are excitatory. So if they interact with the dendrites of the Purkinje cell, what do they do to Purkinje cell?
Excite them.
Excited. And if you excite the Purkinje cell, what will the Purkinje cell do?
Inhibit the deep cerebellar nuclei.
So does it make sense to you guys? Do you see what's happening or you don't? This particular nucleus simultaneously stimulates deep cerebellar nuclei, and through the Purkinje cell, it simultaneously inhibits it, right? Do you get it or you don't get it? Because here we have direct stimulation to the deep cerebellar nuclei. Here we have indirect inhibition. How? Because climbing fibers stimulate the Purkinje cell. Purkinje cell is inhibitory, so it's kind of inhibits the deep cerebellar nuclei. This is fine-tuning. You have two different signals coming simultaneously. As a result, they sort of cancel each other out, right? So you have a movement appearing. There is a very slow movement appearing. So take a look, we have another nucleus that can also do the same. We have a pontine nucleus. Pontine nucleus sends its axons to also stimulate deep cerebellar nuclei. Okay, so this nuclei can be excited by other two nuclei, the inferior olivary nucleus and pontine nucleus. Okay, so these two nuclei, they want the movement to occur. But take a look, pontine nucleus sends additional fibers to stimulate a granule cell. These fibers are called mossy fibers. Mossy fibers are axons from the pontine nucleus. What do? They excite the granule cell. Granule cell sends its axons into the molecular layer. The axons of the granule cell are very special because they split into a T-shaped form. Okay, they're very special, they're specialized axons. Their shape is very weird. Once the T-shaped axons of the granule cell are in the molecular layer, they create parallel fibers. Parallel fibers are axons of the granule cell. And what are these parallel fibers going to do? They are going to synapse with the dendrites of the Purkinje cell. Remind me, granule cell, is it excitatory or is it inhibitory?
Excitatory.
Excitatory. So if granule cell excites the Purkinje cell, what will the Purkinje cell do?
Inhibit the deep cerebellar
Cerebellar
nucleus.
nuclei. So do you see the same process happens here? The pontine nucleus, what does it do? It sends a direct fiber to the deep cerebellar nuclei, and simultaneously through the mossy, through the granule, through the parallel fibers, through the Purkinje cell, it's going to be inhibiting the same nucleus. It simultaneously excites and inhibits it. This is fine tuning, yeah? It's not, you don't tell it, you don't tell them like, okay, do it. You're like, do it or don't do it, do it or don't do it, do it or don't do it. And eventually the movement becomes very, how to say it, it becomes very, very elegant. I do not know how to describe it to you guys. It's a fine tuning process, yeah? Like you find this right spot for the action potential to travel. Because if there was, if it wasn't here, if the brain says, Yulia, pick up this pen, right? And my cerebellum would not be there. My hand will be like, I'm picking up this hand, okay? It's a very, like, it's an action potential. It tells me to pick it up. But what is my cerebellum doing? It's like, pick it up, but don't pick it up. Pick it up, but don't pick it up. And I'm like, slowly pick it up or don't pick it up. And do you see what, I know I'm like stuttering a lot, but you know, do you get the idea? It's not a very precise command. It's like, do it, but don't do it. Yeah, do it, but don't do it. So that's the cerebellum right here. That's cerebellum in a nutshell. That's the fine tuning. So the movement as a result is not very jerky. It's not a command, do it right now. So your arm is like grabbing it and that's it. It's a very slow, very coordinated, very elegant movement because of cerebellum's do it, don't do it pattern. Yeah? Okay, we have additional cells, stellate cell and basket cell. Oh, the parallel fibers are very interesting because they can simultaneously synapse with these three cells, with stellate, basket, and Golgi. So these parallel fibers, they can pretty much synapse with everybody. Okay, so granule cell, it pretty much synapses with everybody. The stellate cell has ability to inhibit Purkinje cell. How? By synapsing with the dendrites. The basket cell also has ability to inhibit Purkinje cell, but it synapses with the cell body. So which one is more efficient at inhibiting Purkinje cell, stellate or basket cell?
Basket.
Basket.
For sure. Okay, so basket cell is more efficient at inhibiting Purkinje cell. If basket cell does its job, if it inhibits Purkinje cell, the movement will occur. Yeah, because if you allow Purkinje cell to do what it wants, it will inhibit the deep cerebellar nuclei. But if basket cell inhibits the Purkinje cell, the movement will occur. Who stimulates basket cell? The parallel fibers. The same parallel fibers which are stimulating the Purkinje cell. So do you see, it's again, do it, don't do it. It's like you're simultaneously stimulating the two opposites. You're simultaneously stimulating the Purkinje cell and the basket cell. The Golgi cell is the regulatory cell because it's the only cell capable of inhibiting the granule cell, the only cell. But guess who tells Golgi cell to inhibit it? The granule cell itself. It's a cycle because the Golgi cell synapses with the parallel fibers of the granule cell. So if granule cell is too active, if it is too active, right, the Golgi cell will be able to inhibit it. There is a threshold, okay? It's all about a threshold. Can you pass that threshold or can you not pass that threshold? There's an area right here, and by the way, I should mention that Golgi cell does not inhibit granule cell directly. It inhibits the synapse between mossy fiber and dendrite of the granule cell. Okay, so the Golgi can inhibit the synapse between mossy fiber and dendrite of the granule cell. So this has a name. So this interaction, so axon of the Golgi, dendrite of the granule, and mossy fiber, this has a name. It is called cerebellar glomerulus. We have glomeruli in the kidneys, and we also have them in the cerebellum. So this is known as the cerebellar glomerulus. So cerebellar glomerulus is just this particular interaction between axon of the Golgi, dendrite of the granule, and mossy fiber itself. Yes? Whew. So do you get what's happening or you don't get what's happening? You need to watch it again, okay? I'll send you a very good video where they actually show you like how the circles go. But the idea is this: do it, don't do it, okay? So that should make it like that. That is the most important part. Just do it, don't do it, okay? So do it, don't do it. It creates this coordination. That is the fine tuning, okay? So this is what you're trying to do. Okay, quickly, so you know what's happening. Which nuclei stimulate deep cerebellar nuclei? Which nuclei excite deep cerebellar nuclei?
Inferior olivary,
And
pontine,
pontine. Through what inferior olivary excites?
Climbing fiber.
Climbing fibers. The fibers that are here, okay, so we will call them mossy fibers, okay? So in the self-control test, so in the exam, they use mossy for both, okay? So not just for these ones, but also for the ones which excite the deep cerebellar nuclei as well. So climbing and mossy fibers, they can excite deep cerebellar nuclei directly. Climbing fibers also synapse with
Purkinje
climbing fibers synapse with the dendrites of the Purkinje cell. Yes, okay. Mossy fibers synapse with dendrites of
Granule cell.
granule cell. Who can inhibit synapse of mossy and granule cell?
Golgi cell.
Golgi. How do you call this whole interaction between them?
Cerebellar glomeruli.
Very good. You call it cerebellar glomerulus. Do you understand that Purkinje cell and granule cell anatomically are upside down to each other, right? They're in the opposite directions to each other because this is the axon of the Purkinje. This is the axon of the granule. They're going in the opposite directions, right? So you could almost say that granule cell is the one which is upside down, right? So it's because the dendrites of the granule cells are here and they're synapsing with the mossy fibers. The dendrites of the Purkinje are in the molecular layer, and they're synapsing with all the different things. If we have to say it kind of this way, which cells can inhibit Purkinje cell directly?
Basket cell.
Yes, and?
Stellate cell.
And stellate. Which cell can inhibit Purkinje cell indirectly?
The parallel fibers.
Parallel fibers are excitatory fibers of the granule cell. So if parallel fibers, they cannot inhibit Purkinje cell, they excite Purkinje cell because granule cell is excitatory. So which cell can inhibit Purkinje cell but indirectly?
inferior olivary nucleus,
Which cell? Which cell can inhibit Purkinje cell indirectly?
Granule cell.
But
Golgi.
granule cell, yes, granule cell excites The Purkinje. So you want to inhibit the granule. And who inhibits the granule?
Golgi.
Golgi. Golgi can indirectly inhibit the Purkinje cell. Okay, so indirectly. How? By simply inhibiting granule cell. Granule cell excites the Purkinje cell through the parallel fibers, right? Okay, this is like an overview of what we have talked about. Okay, so just spend some additional time there. This is the same. We're just gonna add a little bit of the details right now. So Purkinje cell is the most important cell in the cerebellum. It is a GABAergic neuron. It's inhibitory. Typically, the more inhibited cell is, the more dendritic spines it will have. This is a very important part. Do you see there are lots of these things right here? So these are dendritic spines. Purkinje cell is considered to be the most inhibitory neuron there is, because the amount of these spines that it has, it's crazy. Okay, so it has lots and lots and lots of spines there. Now, parallel fibers, you remember, these are axons of the granule cells, right? Climbing fibers we have talked about. Don't forget glutaminergic, okay? So not acetylcholine. Acetylcholine is for the brain, for the cerebrum. In the cerebellum, we don't use acetylcholine, we use glutamate. Okay, basket and stellate cells, we have talked about them. They are inhibitory, so they must be GABAergic as well. Purkinje cells, what is their function? They regulate and coordinate motor movements. I just want to add additional sentence to kind of make it a bit more clear. How exactly Purkinje cell inhibits? Because you have to understand that Purkinje cell is the only cell that can inhibit deep cerebellar nucleus. Do you see the rest of them excite? It's the only cell that can inhibit. So try to understand it this way. There's so many excitatory axons coming to this nucleus. If there's so many excitatory axons coming here, agree with me, they are firing too many action potentials at this nucleus, right? And if there are too many action potentials, that will cause an additional stimulation of the motor neurons. So the contraction will be very rigid. But if Purkinje cell has a little bit of the inhibition adding to it, it's like you're reducing the strength of contraction. Purkinje cell alone cannot inhibit it. Okay, so do you know what I mean? It cannot just stop the movement completely. There are hundreds of excitatory inputs to this nucleus and one inhibitory to this nucleus. Who do you think is going to win? Excitatory 100 or one inhibitory? Who is going to win? Of course, excitatory. But will it be as strong? It won't be. That's the job of the cerebellum. It makes it more coordinated. So when you reach for something, when you grab for something, your movements are very smooth. Yeah, they're very coordinated. That's the job of the Purkinje, because it tries to suppress this overexcitement. It's like, think about it this way. Your brain sees a cap and he goes like, oh my God, I want that cap, like must get it now. And the Purkinje is like, cool, yeah, like chill, well, let's just reach for it nicely. It provides this opposite force to it. So your neurons aren't fired up to get that cup. Yeah, they're like, okay, we will get it. So does it make sense more or less?
Julia,
Oh,
what excites the pontine nucleus and the inferior olivary?
very good question. We'll talk about it next class.
Okay.
The brain. Okay, so the brain. Yes, the brain. Okay, this is important. Which deep cerebellar nuclei, Vera, right? I told you there are 4 of them here. Which ones, Vera? dentate, emboliform, globose, and fastigiae. Okay, so these 4 are the nuclei present in the deep cerebellar region. They are the deep cerebellar nuclei. Okay, granule cell. You remember the excitatory T-shape, blah, blah, blah. This is important. It possesses dendritic claw. And typically it only has 4 to 5 of the dendrites. So do you see, this is your granule cell. This is the cell body of the granule cell. These are the, how do you call, the specialized axons. Okay, these are the specialized axons which are going up, splitting into the T shape, right, and creating parallel fibers. You can see that these are 3 to 4 dendrites of the granule cell. In the dendritic claw, into it, the mossy fiber comes in. But who can inhibit this interaction?
Golgi cell.
Very good. Okay, Golgi cell. Together, this is known as cerebellar glomerulus, right? So if we have to specify, cerebellar glomerulus is basically mossy fiber, dendritic claw of the granule cell, and axon of the Golgi. Yes? Yeah. Okay, we talked about it. This is just for you additional information. So when you study, just read it at home, yeah? Okay, so stellate, basket, you remember. Which one has axosomatic synapse?
Basket?
Basket, yeah, because basket synapses with the cell body. So axosomatic synapse is done by the basket cells. Okay, so the same guys we talked about it is just additional information all over again. Yeah, so just what we have mentioned previously on that one picture. Okay, it's just here. So when you're studying, just it's the same idea here. Okay, mossy fibers. It's one of the major inputs into the cerebellum. Mossy fibers originate from the pontine nucleus. Mossy fibers do not only come from the pontine nucleus. There are other nuclei who also send mossy fibers. This is going to be vestibular nucleus, spinal cord, and reticular formation. Okay, so these mossy fibers do not only come from the pontine nucleus. So is it correct to say that mossy fibers, okay, before that, which peduncle is interacting with the cerebellum and pontine and pons?
Middle.
Middle. So is it correct to say that mossy fibers can only be present in the middle cerebellar peduncle?
No.
No, because you have to remember that mossy fibers do not only come from the pons, they also come from the spinal cord, they also come from the vestibule, right, and etc. So these mossy fibers can also be present in the inferior cerebellar peduncle as well, because spinal cord, right? You remember? Spinal cord sends its axons through the inferior cerebellar peduncle, right? Okay. So of course you have to understand that they're coming into the cerebellum, so it has to be sensory. So these mossy fibers, they just sort of carry the sensory information about your general position in space, right? So vestibular, sorry, that's your balance. Spinal cord, that's how your muscles and joints are. So these are sensory fibers. They're just making sure that cerebellum is aware of your position in space. Okay, climbing fibers are coming from the inferior olivary nucleus. Inferior olivary nucleus is located in the medulla oblongata, and I told you that medulla oblongata is going to be sending its axons through the inferior cerebellar peduncle, right? So that's where the axons will be. Each climbing fiber can synapse with 1 to 10 of the Purkinje cells. Okay, so there are lots and lots of Purkinje cells that can synapse with it. There are additional fibers, but they are inhibitory because MOSI and Clybin are excitatory, right? They're inhibitory fibers. They come from Raphe nucleus, and they are monoaminergic fibers. Okay, so about Raphe nucleus, we will talk about it, but honestly, it's not as important because the major input for the cerebellum are MOSI and Clybin. They just asked this question like last year to make sure that you remember that that's not the only input into the cerebellum. There's additional input from the nucleus of Raphe, and it is inhibitory. This is a very common question. What is the output of cerebellum? What's the only output of cerebellum? It's going to be axon of Purkinje. Okay, that's the only output. So that's the information that leaves the cerebellum. Okay, through the axon of Purkinje. Where does the axon of Purkinje synapse? Deep cerebellar nuclei or lateral vestibular nuclei. That's why cerebellum has something to do with the balance as well. So not just controlling the movement, but also balance. Okay, this is the microcircuit. Read it at home. It's everything that we have talked about before. This is a very important slide because here we're discussing some kind of terms that are very useful, not just to pass the neuro, but for you as a doctor. Okay, so this is very important right here. Basically, which kind of symptoms will occur if you have damage to cerebellum, okay? If there is something wrong with the Purkinje or, I don't know, any other neuron, right? So what could be seen? The most important symptom is ataxia. Whenever you see ataxia, this is something with cerebellum. Ataxia is abnormal gait and uncoordinated movements. Ataxic person always tilts in certain direction. It's like you ask them to walk in the straight line, and if your cerebellum is fine, you will walk in the straight line like nothing is wrong. Ataxic person will be tilting, and it depends. Okay, that's where, please don't make me go back, that's where this particular horseshoe comes in. Where's the horseshoe? I skipped it. Okay, so that's where the horseshoe comes in. Because you remember, it depends on the lesions. Because if the lesions are going to be made above it or below it, it depends. If the lesion is above it, if the patient is tilting in the wrong direction, into the left, that means the damage is on the right side. But it also means that if the damage is below it, if he tilts to the left, that means that the damage is on the left. So that's why in cerebellar damages, we don't really pay attention to ipsi or contralateral, okay? It's not important. In the cerebral damage, it's important because it's always crossing. In the brain, it's always contralateral. But in the cerebellum, do you see, it could create ipsilateral as well. So we don't care about the side that this is happening. It's not a very important symptom. If symptom is happening, if ataxia is already happening, if the person is already tilting to the wrong side, it's already cerebellar damage. That's it. So we don't care about which side is it. It's already enough to know that this is cerebellar damage. Does it make sense? Okay, so that's ataxia. Dysmetria, misjudged distance. You put a chair in front of them and tell them to stop in front of the chair. They won't. They will always bump into that chair, always. And it's, I know it's bad, but it's always funny to watch how they realize that they bumped into chair because they haven't expected that, okay? So in their brain, there is still a bit of a distance between them and chair, and then suddenly this chair appears, so they're so like surprised. I guess this one is the most specific for cerebellum, the intention tremor. Tremor, as you know, is shaking, right? Like shaking of the hands or something like this. Intention tremor, it means that whenever you want to grab something, right? I want to grab this glass right now. The tremor gets worse as I approach the glass because I'm intending to grab this glass, but I can't, yeah? Or like you tell them, I don't know, button your shirt up, yeah? So they lift their arms normally, but as they approach the button, it gets more and more tremory because they're intending to do it, yeah? This is the intention tremor. Very interestingly, it's not only with the hands, it's also with the head. You tell them to turn their head to the wrong side, like to the left side, for example, by saying, look at me, yeah? And as they turn, as they're trying to reach you, it gets worse and worse and worse. Why? Because the intention is done by your brain. Your brain is like, pick up this glass, okay? Pick it up now. But cerebellum is damaged. Purkinje is like, yeah, do it. Like, I can't do anything, yeah? Like, I can't inhibit, do it. So do you see, it gets worse. The tremor gets worse because the brain keeps on firing that you must take this glass right now. But Purkinje is not doing its job. It's not calming this action potential down. Because cerebellum has something to do with the vestibular, you see Purkinje synapses with the lateral vestibular nucleus. It's all about balance, right? You're going to see that there will be vestibular signs. There will be nystagmus, there will be head tilt. Okay, this is a head, very wrong, weird. Okay, so head tilt, they tilt their head so they can stabilize the balance, right? Anybody knows what nystagmus is? No? It's when the eye goes, it starts moving. Do you know what I mean? It's not a lazy eye because lazy eye is lazy, okay? So it just stays on one side. But you see that the eye does this, okay? The eye does this movement. Have you ever seen it? No? Okay, so YouTube it, okay? So nystagmus is, the person is looking at you, but you can see that the eye moves just a little bit, okay? So it just moves just a little bit. So this is going to be the vestibular sign. Okay. So let's take a look at the cerebellum. Okay, so these are the folia. Okay, so you should know that these are the folia right here. So if I go back, the folia are the gyri of the cerebellum. This is the white matter, and this is the gray matter right here. Right, so the white matter is the linea alba, lamina alba, sorry, and this is the area where the nuclei will be. So this is the gray matter, right? And this area right here is the white matter. And then again, gray matter continues. What is this?
Pia matter?
Yeah, for sure. Yes, this is pia matter. Yeah. Okay, so this is pia matter right here. So how do we call the outer layer?
Molecule?
Very good. Which neurons are going to be there?
Basket and stellate.
Yes. Okay. Anything else is here?
Dendrites of the
Of the
Purkinje.
Purkinje and
Parallel fibers.
parallel fibers. Very good. Okay, so parallel fibers are also going to be here. The second layer right here, this is the Purkinje layer, but as you can see, we don't really see much. We just see the cell bodies of the Purkinje. Okay, so here they are. They're just huge cell bodies of the Purkinje. And then the last one is the granule layer. That's where the Golgi and the granule cells will be. Right, so this is going to be the pia matter. It's important to remember that in the cerebellum or surrounding the cerebellum, pia matter is vascularized. It's super weird. Okay, so typically in the blood vessels could be in the subarachnoid space or something like that, but in cerebellum you can see blood vessels there. Okay, so blood vessels are present in the pia matter. Okay, so these are the cell bodies of the Purkinje cells, right? These nuclei probably belong to the basket and stellate cells. And the lower layer, this is going to be the granule layer. Do you see that? Yes. Do you see that we have some kind of empty spaces here, right? So these little tiny empty spaces in the granular layer. So we magnify to them. This is cerebellar glomerulus. So this particular space, okay, so this is the cerebellar glomerulus. Remind me what is happening in the cerebellar glomerulus?
The dendrites of the granule cells,
Mm-hmm.
the axons of the Golgi cells, and the mossy fibers.
Very good. Yeah. So it's the three things, right? So it's better said dendritic claw, yeah, because granule cells are very specific for that. They're known for that there is a claw here. This claw interacts with the mossy fiber, and the axon of the Golgi will be inhibiting that. So it happens right here. Okay. So in the space, that's where you'll find those things. Okay, so the same idea. So vascularized pure matter, molecular layer, Purkinje granule layer. You can see cerebellar glomeruli, right, and etc. Bielszowski impregnation. You should know that they typically point here. Okay, so you can see this area. They typically point here and ask you, what is that? This is lamina alba. Okay, so it's just to confuse you because it doesn't look white. So you would never choose that this is lamina alba, but you know, it is. Okay, so this is the white matter right here. This is lamina alba. On the surface, you can see that there are a bunch of meninges, right? So probably majority of it is pia matter, right? And it is vascularized pia matter as we have discussed it. So this is Bielszowski, so it is silver. So silver will show you axons in black, cytoplasm in yellow. Pia matter will be visible very nicely. You see, black color is evident here because pia matter possesses reticular fibers. Molecular layer will possess black dots. These black dots will be the cross sections of the parallel fibers. Okay, so take a look. This is the molecular layer. This is the granule layer, sorry, Purkinje layer, and this is the granule layer. This blackish area here, this is lamina alba, right? So this is where the white matter is. So they always point here, and that's where you get confused. This is a cell body of the Purkinje cell, right? These empty spaces right here, these are going to be the cerebellar glomeruli. In the molecular layer, these black dots are parallel fibers. They're cross sections of the parallel fibers. Yeah. Want to get your attention to here, right? So you can see that this is the Purkinje cell, and you can see that there is a cell here that sends its axons to the cell body of the Purkinje cell. So which cells are these?
The basket cells?
Mm-hmm. But which layer is this?
The granule.
Yes, this is a granule layer. Is this a normal position for basket cells?
No.
Surprisingly, yes. In theory, basket cells are in the molecular layer. In reality, basket cells are in the granule layer. Okay, so according to the books, according to the lectures, according to all of this, you could find the basket cells only in the molecular layer, but that's actually not the case. Okay, so you will actually find them in the granular layer. What are these dots?
Cross section of the parallel fibers.
Very good. What are these lines?
Dendrites
Yes, whose dendrites?
of Purkinje cells.
Purkinje cell, yeah. So these are the dendrites of the Purkinje cell right here. Golgi, you remember it's just extra silver, okay? So double the silver. So what do we see? I guess the purpose of this slide is to show you the Purkinje, how the dendrites of the Purkinje look like, right? So these are the Purkinje cells right here. and their dendrites are quite extensive. Okay, so if you take a look at this area right here, so you can see that these are the dendrites of the Purkinje cell. Okay. So yes, okay, so these are going to be the axons, the dendrites of the Purkinje cell. Notice how many spines, do you see? It has lots of little tiny, like almost hairs that are visible here, right? So this is the axons of the dendrites of the Purkinje cells, okay? So the dendrites of the Purkinje cells, and you can see that something is climbing up. Okay, so this is actually a climbing fiber. It's the axon, the climbing fiber that is going to synapse with these dendrites right there. Okay, so the same idea. These are the dendrites of the Purkinje cell. Look how spiny they are. Okay, they're very spiny. So don't forget that Purkinje cell is the most inhibitory neuron in our body. And you can see this. What do you think this is?
The dendritic claw.
Yes, this is a granule cell, and this is the dendritic claw right here. You can see 3 dendrites are visible, right? So 3, typically it's 3 to 4 dendrites, 4 to 5, but at the end, you should always see dendritic claw. Okay, guys, I forgot the name of this cell. I will get back to you. Something Bergman. It's a very special type of the astrocyte in the cerebellum. Okay, so they always ask this question. I will get back to you next class. I need to open my notes, my other notes. Okay, because I did not prepare for today. So this is just additional specialized astrocyte in the cerebellum, and this is how it looks like. Its axons are in one line. Okay, so it's like one, I don't know, huge brick of axons that are coming out like that. It's Bergman something. I remember Bergman, but the first part starts with K, and I don't remember. Something Bergman cell. Yeah.
Yeah, it's Bergman cell.
Yeah. Okay, but there's additional name, and I need to open my notes for that because I don't remember. Yeah. So Bergman something. Yes, Bergman. Okay, that's it for today's. Okay, so yeah, Bergman. There you go. Yes. But,
Julia, we can't see the inferior olivary nuclei cell bodies.
Inferior olivary nuclei are in the medulla oblongata. So we need a section of medulla. This is cerebellum. Okay, so the same why we cannot see the pons nucleus, because it is in the pons. So all of these nuclei that we have talked about, we definitely cannot see them. We will only be able to see maybe some fibers, but we just saw climbing fibers, yeah, and even MOSI weren't really visible, you know, but yeah. But actually, let me show you something, because I think it's important. I remember there was a question there once, many years ago. They pointed here, right? So not here. Yes, okay, so they pointed here at the lamina alba. And do you see that there are lots of lines here in lamina alba, right? So there are lots. Can you see my screen? So in the lamina alba, there are lots and lots of lines. So they pointed and they said, hypothetically, which fibers could those be? The answer was MOSI. Okay, so hypothetically, the fibers that you see in the lamina alba, they could be MOSI because they're coming to synapse with the deep cerebellar nuclei. But yeah. Okay, guys, so that's it for today.